Okay, welcome everyone to the third session of the 2026 Spring Series on magnetic fields in biology. And it's a real delight to have Wendy Beane back again speaking to us. So thank you so much for coming on today, Wendy. And you're going to be introduced today by Sanika Ghayal, who is a PhD student at the School of Life Sciences and the Guy Foundation Quantum Biology and Bioenergetics Laboratory at the University of Westminster in London. And she'll be introducing the topic and then introducing you, Wendy.
Thank you, Geoffrey. So, good morning and good afternoon, everyone joining us from different time zones. I'm Sanika Ghayal. I'm a PhD student here at the University of Westminster, and I'm very pleased to be here today in this introductory talk in the spring series on magnetic field and biology. So, today's topic is the evidence for magnetobiology in animals and humans. So, before going to our main topic, I would like to give a quick recap about the previous session, where we heard Professor Margaret Egg and Professor Martin Shah, who gave us a talk about the role of magnetic field in um, therapeutics. For example, in therapeutic nuclear resonance, where the relatively low magnetic field was applied in a controlled condition, was shown to be influence the cellular processes such as ROS signaling, effects on metabolism, tissue repair, and also it showed potential applications in wound healing and musculoskeletal conditions. In contrast, an oscillating magnetic field, where the magnetic exposure changes over the time, uh, found to be modulate cellular behavior. And it also affects on the oxidative pathways, ROS signaling. And also it was important in cancer where it was shown to influence various tumor-related pathways. And now it has been further investigated for its therapeutic applications. So basically, the last session highlighted the biological effects basically, and how the strength of magnetic field and how it is applied affects the whole different pathways and biological processes. So now, which brings us to our main topic today, which is magnetobiology. So on Earth, we are all exposed to geomagnetic field, which ranges from 25 to 65 microtesla, and it has shown to affect the various biological processes from nuclear molecular level to whole organism level. So at molecular level, it affects the ROS signaling, the other proposed mechanism, which was previously also discussed in this series, which is radical pair mechanism, where the outcome of the reaction basically depends on the spin state um, of the electron, and it has shown to influence by the magnetic field. At cellular level, it affects the cell proliferation, differentiation, and at whole organism level, it affects the orientation, circadian rhythm, cardiovascular effects, migration in birds. So overall, magnetic field is just important in influencing various biological processes. So now moving on, and it becomes very relevant when we think about the magnetic fields beyond Earth. When we talk about the space environment where there is no magnetic field, and missions like Artemis, where it marks a key step towards long-term space missions to Moon and Mars, the main question arises, which is what happens to biological systems in the absence of geomagnetic field? So this review basically summarizes the effect of hypermagnetic field in vivo and in vitro. So in vivo, it affects the physiological functions, which are cardiovascular systems, neurocognitive functions, musculoskeletal functions, circadian rhythm, reproductive function. And in vitro, it affects gene expression, mitochondrial function, metabolism, cell proliferation, differentiation. So hypermagnetic field is not just only affecting one process or one pathway, but it is affecting different biological processes. And this brings me to my next slide, where I would like to present some of our work here at the University of Westminster as a part of Guy Foundation Space Health Program. So in this image, this is the hypermagnetic chamber we use here at university, and it is made up of a special alloy called mu metal. And at Westminster, we assess the effects of hypermagnetic field exposure in vitro. So past 1 year, I've been working on THP1 immune cells, which are human monocyte cells. And as a part of my PhD, I'm working on C20A4 chondrocytes, which are cartilage cells. And now I would move on to our main topic, where I would like to introduce our today's speaker, who is Professor Wendy Beane. She is from Western Michigan University, and she has done her PhD in cellular and developmental biology from Duke University. Her main research focuses on regeneration, bioelectric signaling, and quantum biology. She has 30 plus publications and over 3,000 citations. She recently received 2024 Excellence in Research Award. And apart from that, she leads various quantum biology initiatives, such as she coaches the Gordon Research Conference. She's a part of US Quantum Biology Center. And beyond laboratory, she's very passionate about STEM outreach programs where she introduces students about regeneration and sparks curiosity through real experimental science. So over to you, Wendy. Thank you.
Thank you so much. I'm really glad to be back here and hear a little bit about Sanika's research, which seems very interesting. Today, I'm going to talk to you a little bit about biological effects of magnetic fields on animals, which includes humans. Which on the face of it seems pretty straightforward, but it quickly got very complicated. And so I decided to take a little different tact this time, and more so walk through why, when we look at the effects experimentally, right, in the literature, they're a little bit all over the place, right? You get a lot of different systems, you get different results in different places. And I thought it would be worth a discussion, just sort of outline sort of why, right? Or at least why I think why. So, um, the first part is the sort of biologically relevant magnetic field processes, I guess. There's obviously magnetoreception, migration, orientation, that sort of thing. There are endogenous bioelectric magnetic fields, so you can, there are small magnetic fields generated from the heart, the brain, across cell membranes. And what we were talking about a little bit earlier, there is a really kind of exciting new area of research showing that cells, bacterial cells, but also cells in multicellular organisms like animals and humans, appear to be using biomagnetic fields in a way, or wavelengths in a way to communicate with each other, and in group oriented ways. I love uh, this is from the Beane Lab at UC San Diego, and he calls them biological cell phones, which I think is great because it's kind of radio waves. All right. The ones I study in my lab are another whole different area of sort of magnetobiology, which is the effects of external magnetic fields outside the organism, and specifically what that exposure does in cellular and molecular effects, right? So all right, so we can narrow it down to maybe external fields and leave the others for another day. But that doesn't actually make things a little bit easier, because what we mean by exposure is kind of all over the place in the literature, which is both frustrating and really, really exciting, right? So I think as we get more people studying this in each specific area, it will be very nice to have all of these, but you kind of have to not just say what are the magnetic field effects, you sort of have to figure out what you're dealing with. So what I have been traditionally studying, and a lot of people study, are static weak magnetic fields. The problem with that, even that, usually they're quote, weak, which in our lab we arbitrarily call anything under 1 millitesla. It can vary up to less than 10 millitesla or 1 tesla. It just depends on whose definition of weak. Statics usually the same definition, fortunately. The other large percentage of the data are these extremely low frequency electromagnetic fields, the ELF EMFs. And they have some really exciting cellular effects, particularly at frequencies that have physiological relevance. So most of them seem to be less than 200 and 300 hertz, anywhere from 0 to in that range. But the most ones that I see from sort of an animal human point of view are around 5 or 15 hertz, and especially 50 or 60 hertz, which debatably might have to do with calcium flux, which will come up in a second. There are also some other ways that human tissues and animals are being exposed. More in like artificial settings, so the hypomagnetic fields that we heard about, you kind of have to generate them on Earth, right? So we need to go out to space to find them or create them here. That's typically in the range of anything less than geomagnetic, though I think it's starting to mean near zero, but we'll see. Then there are some things that are already being used as therapies that have already probably been talked about, sort of pulsed EMF fields. Those are particularly used in orthopedics. They have very good effects on bone formation and growth and collagen containing tissues. So they're used for treatment for lots of things, including bone bruises and tendonitis and that sort of thing. But you get slightly different results than you do with the other types of exposures. And the same for the sort of rotating permanent magnet setups in the transcranial magnetic stimulation therapies. Those are typically being used somewhat to really good effect, especially with pain, for nerve-related issues. So chronic pain, depression, and some other neurological situations. All right. So if we just narrow it down, right? Okay, I'm just going to look at static weak magnetic fields and maybe the frequency, right? The EMF. Um, and then I'm only going to look at cellular molecular effects. We're not going to worry about pretty much anything. That also does not narrow it down as much as it should. And I think right now this is a confounding factor because we are trying to learn a lot of foundational information about how magnetic field exposure is affecting basic cellular functions, right? Like how that works and how it works in different situations, right? But I think in over time, the fact that it affects the things we're about to talk about, which are kind of broadly applicable to a lot of animal biology, will actually be a strength. So the main sort of three characters or suspects that can be traced to the effects from magnetic field exposures, particularly statics and the EMFs, are changes in ion flux, which by definition means you're changing cellular membrane voltages. Magnetic fields are very good at that. Free radical formation and dynamics, sort of electron spin, hyperfine interactions, that sort of thing. And there is the cytoskeletal data is very interesting. The experiments are very interesting. You definitely get cytoskeletal changes in microtubules and actin polymerization. There's a couple ways that you can do that. It can directly affect the polymerization dynamics of the tubulin and actin, but it also can be upstream of the cell signaling that controls that. In fact, all three of these things basically feed into cellular signaling, right? And the way that evolution sort of has created sort of eukaryotic biology is it kind of uses the same mechanisms in different places for different effects, right? So you could take the same thing, the same cellular signal, biochemical ligand, whatever protein, and you'll get different effects at different times in different places in different tissues, right? So a lot of confounding factors. So it's going to take a lot of looking at things individually, right? So I think having sort of a better understanding of exactly how these three areas that magnetic fields are very good at producing significant and relevant effects in animal tissues at the cellular level um, um, will sort of help us navigate how we go forward and figure out the sometimes contradictory data that we see in the literature so far. All right. So the first target, actually, I'm starting with the last one, the cytoskeleton. That cytoskeletal dynamics have a lot to do with everything. But the two probably most relevant, if we're going to talk about changes at the tissue and organismal level, or organ level, maybe, are the cytoskeletal dynamics drive cell migration. Right. So where cells migrate, how they migrate, and when they migrate. And this also feeds into signal transduction. So the cytoskeletal dynamics can actually change the rigidity of both the plasma membrane and the matrix that the cells sit on, right? And those are well-defined and known signaling mechanisms. So mechanosensory receptors, you have signaling molecules that are being diffused and carried through the extracellular matrix. So by messing with cytoskeletal dynamics, you can really change significantly the signals that individual cells are receiving or giving. The cytoskeleton also, of course, has a lot to do with cell division, both microtubules and actin separately. And probably maybe therapeutically, the most interesting would be epithelial to mesenchymal transition. So when cells in development, uh, during embryogenesis, when we go from a single ball of cells, right, like a sort of a hollow single layer ball of cells, and we're trying to form all of our different germ layers to make all the different tissues. The mesenchyme, or the tissues that are going to sort of end up in the middle, right, between your skin and your linings, like your gut lining and your um, uh, vascular linings, right? They undergo this process where they come out of this epithelium, which is your knit tight group in your skins, and they migrate individually to where they need to go on the inside of the embryo. This is a direct mechanism that tumor cells use to undergo metastasis. So it becomes, if magnetic fields are going to, and they do, affect cytoskeletal mechanics and dynamics, that will have direct relations to affecting the activity of tumor cells, right? So all of these things are things that there is evidence for in the literature, in human cell culture, some ongoing therapies, but not most so much the stuff I'm talking about, and in animal models. Okay. All right. Probably the big one is ion transport. The magnetic fields have several things that they can do in several different ways that they can affect ion flux, which means that we're dealing with membrane voltage. From a sort of cell biology or a developmental sense from embryogenesis, what we are interested in is actually membrane depolarization, which just means the inside of the cell has more positive ions than the outside of the cell. All right, so you can sort of see here this is sort of an illustration, I guess, of a quick action potential when some positive ions are going through the plasma membrane and doing a quick depolarization. Okay. So the thing to keep in mind is that this happens through ion transport proteins that are in the membrane, plasma membrane. And this membrane voltage is just literally, the ion concentration inside versus the outside of the cell. At resting state, the inside of cells are usually more negative than the outside. And depolarization is a biological way to activate a biological process. Okay. And frequently, a lot of times, this has to do with calcium entry. Calcium flux is super important in pretty much everything we're going to talk about, but the ion transport that magnetic fields can affect is not just calcium. All right. So the ones we think of when we think of changing membrane voltage are these quick action potentials in sort of excitable cells that you sort of learned in physiology, right? So your neural active, your sort of the function of your nervous system, the depolarization of those quick millisecond depolarization events that conduct nerve impulses. The same sort of action potentials occur in your heart, and you also get see depolarization at sort of neural muscular junctions, right? So instead of nerve to nerve synapses where the nerves innervate the muscle tissues. Now, all of these are very quick, where the resting state is in the inside is hyperpolarized, so it's quite negative. And then there's a quick depolarization event where ions come in and then it resets, it goes back, right? But the thing is, all cells, this is me showing my Mike Levin postdoc background. It all comes back, right? Everything you study ties into anything else you study. Um, it, so we traditionally think, at least when I was back in the dark ages, a graduate student, we typically thought of excitable cells as having membrane voltage being super important, and every other cell, not really, right? Because we're sort of taught that all cells basically have a resting potential that is pretty much the same, and it's these excitable cells that go through the depolarization. Except that's not true. All cells undergo, I went too far, there we go, undergo changes in their membrane voltage. One of the most powerful times is actually during developmental processes. So development we tend to equate with embryogenesis, and a lot of the things I'm going to show you here are during fertilization and embryogenesis, when changes in membrane voltage, specifically depolarization events, are super important for driving these. So if magnetic fields are changing these, which they do, um, that's going to have consequences, or we'll be able to use therapeutically, hopefully, on purpose, right? But developmental is sort of basically anything that happens to you in your entire whole life, including aging. So there are some really interesting things, like in our lab, we study new tissue growth sort of as tissue repair downstream of stem cell mediated regeneration. And that is a developmental process, and it does rely on bioelectrical signaling with changes in ion transport and membrane depolarization. So if I can get this to go, what you see here is just one example. There's more of how membrane voltage is really, really important during fertilization. The sperm binding with the oocyte actually causes a wave of membrane depolarization that leads to these really quick transient calcium fluxes that actually put up this sort of fertilization envelope, which acts as a block to polyspermy, keeping other sperm from bringing more chromosomes to the party and disrupting the downstream organisms. But pretty much all throughout embryogenesis, like control of tissues membrane voltage is very important for the proper development of the organism. And these changes are not at the level of milliseconds, right? They can be minutes, days, hours, and some of them are steady state, kind of permanent, especially like in adult planarians. We just have some that are just always differentially maintained in potentials. Here is an example in Xenopus, where you can see now instead of looking at depolarization, we're looking at hyperpolarization in the lighter areas, just kind of a reversed image. And what you're seeing is the nervous system coming together in these tadpoles and all the craniofacial features where you can sort of tell where the eyes are going to be by these bright hyperpolarized spots, which actually denote the eye fields before any of the biochemical protein markers of the eye fields are found. So all throughout embryogenesis, membrane depolarization events control a lot of those activities. Here we go. And even later, this is not working. Come on. Okay. Um, later in embryogenesis, so I guess it should have been part of embryogenesis, but specifically, even as we get further along in development, like the final organization of our organs are also depending on these ion transport and membrane uh, voltage, right? So in this case, hyperpolarization is something that increases the size of organs in several different contexts here. It's increasing it in Xenopus tails. And even more interesting, they have found, we're back to Xenopus here, the frogs, that if you ectopically hyperpolarize, I told you about those little dots of the hyperpolarized eye spots. If you ectopically hyperpolarize, it's kind of hard to see here, but there is an innervated, what we think is actual functional eye, ectopic eye, that comes up just because that tissue membrane voltage was changed. So it's a very powerful target that has a lot of real world effects. Okay. Now, tied in with membrane voltage is ion flux, in this case, specifically calcium flux. So, in terms of cellular processes and development of embryos and sort of normal cellular homeostasis, calcium has a very kind of special place among most of the ions. It functions as a second messenger on its own to amplify signals that are coming extracellularly from a cell. So during cell communication from its environment and between other cells. So the membrane voltage that we're interested in, there are voltage gated calcium channels. So they are calcium channels that sit in the plasma membrane and they are gated, so they're closed. So calcium cannot change. But when the membrane undergoes a depolarization event, they open and calcium's allowed to travel in or out on its gradient, right? Which is usually in because the resting state is usually negative, right? So you get at many critical events during development, during fertilization, embryogenesis, and wound healing and regeneration, you get a membrane depolarization event that is quickly followed by a calcium influx. So in the cells that we study, there's a burst of membrane depolarization at the wound site after injury, which is followed by and required for the burst of calcium at the wound site that serves as part of the injury signal after injury. Calcium signaling is so important that there are actually two stores inside every single cell in the plasmatic reticulum and the mitochondria. Both store. The mitochondria more of a backup in case something happens with the ER. But given that we've shown that magnetic fields can change calcium flux, and calcium is known to be upstream of things like whether a cell undergoes cell death, whether it divides, what stage it is in the cell cycle, the ability of your nervous system to adapt, its plasticity, the migration and invasion, also that we talked about because cell adhesion is calcium dependent, and a lot of other things require calcium. So these are all sort of areas where the ramifications of the fact that the magnetic fields have been shown time and time again to be able to control, right, the cytoskeleton and the membrane voltage and the ion flux, particularly the calcium, um, means that we are going to get a lot of effects depending on where we are exposing and what tissue and what system we're in. Um, uh, and part of the reason that everything is sort of all over the place, and if you're trying to look at what the effects are, is because of the way the organism is evolved. The cell is like the world's best recycler. It's not going to come up with a new mechanism if it's already got one, right? So if it's got some signal it used during development, it's like, hey, why don't we use it for, I don't know, immune system or something, like we just going to use what they have lying around, right? It's, I guess, efficient, right? And so all of these things, the depolarization and the calcium and the cytosolical changes, cause different downstream effects depending on what tissue they're in and what time they're in, right? So the age of the cell or the age of the organism or circadian rhythms, the literal time of the outside world, that sort of thing. From that point of view, the next big one is radical oxygen species, which is why I left it to the end. So in addition to everything we just talked about, we have shown that magnetic fields are able to change the levels of radicals in the cell in a biologically important way. So I'm pretty sure most people know reactive oxygen species have a free electron that's looking for a home. I leave this here because biologically, probably the two most important are the super oxide radical and its derivative hydrogen peroxide. Especially hydrogen peroxide is a very large signaling molecule. It's also at larger levels toxic, but it has a lot of roles, as we will see. So when we start getting into, even on top of everything that the ion flux was doing, what ROS is doing as a signaling molecule, right? So we now get all over the place, right? So during maturation of the sperm, right, there is a certain level of reactive oxygen species, or ROS, that are required for the sperm to mature and be able to fertilize the egg. However, if you get at other levels, usually much, much higher, that then stops being something that is physiologically helpful and starts being something that is definitely pathologically not helpful. All right. We see some of the same when we look at the immune system. The graph I have here is for the innate immune system, but ROS is as the nice intro explained, is also very important for adaptive immunity, especially like T cell function and cytokines and all that kind of stuff. But again, you have levels of ROS, times and amounts of ROS that are very helpful for immunity, right? To help us fight invaders and that sort of stuff, even get rid of tumor cells, that sort of thing. And then we have levels where it becomes very, very harmful. Okay. We see this trend again and again. Here, we're talking about the role of reactive oxygen species in tumor progression, right? So there is a role for ROSs, which are increased during tumor proliferation, right? But they're kept at a level that's not too high, because now we've got our harmful and our beneficial split, right? Because a signaling level of ROS is good for the tumor cells to keep dividing, but they don't want them to get to levels where we are going to turn on tumor suppressor or cell death pathways, which then would become beneficial for the organism as a whole. In a million, I could just keep going on, but I think these, especially the last three, but uh, these illustrations are probably maybe the most interesting. There is some really good data showing that ROS plays a role in neuroinflammation, and it has again to do with concentration and timing, right? So if you're going to fight an infection, right, you want a high anti-inflammatory state early on with large ROS levels, and then later you want that to die down. And if it doesn't, it then becomes harmful, right? And these are things that, except for the sperm maturation, I couldn't find any data on that, but these other three areas are things that the research has shown magnetic fields are able to affect these processes, right? Either in tissue culture or in animal models, right? Um, so um, what I want to kind of narrow this down to is something that we are focusing on in our lab now as a sort of a natural product of sort of our research up to date, is there appears to be sort of this bioelectrical ROS signaling pathway that is very important for tissue growth and tissue repair, and probably several other mechanisms, but that's what I study and I know the most. ROS is not on this chart, unfortunately. It would be right here. But what we have is a membrane depolarization event that opens those voltage gated calcium channels. That leads to downstream ROS production. But it's a kind of interesting loop because ROS is both downstream of calcium and depolarization and upstream of it. There is a feedback loop specifically with this MAPK signaling pathway. ERK, RAF-RAS, and the ERK, which is the MAP kinase at the end that goes in and controls the transcription and gene expression changes that specifically cells that are dividing, right, need in order to proliferate. Okay, so this is why they get turned on after injury. It's why the cancer cells want them turned on so they could keep dividing. And it makes it a good target both for regenerative medicine as well as um, oncology, right? So, but there is a timing mechanism, especially during wound repair, like we saw with neuroinflammation, where you want it on, and then at some point you want the ROS and the depolarization in calcium off, right? So this feedback loop that we're starting to investigate is a way to both amplify the signal, keep it on for the time amount it needs, and then at some point you will be able to interfere with that, and then everything will fall apart and you will stop the signaling, right? So this this sort of depolarization bioelectrical signaling pathway appears to be very close to the injury signal. Well, talk about that in a minute. Okay. So why all of this different mechanisms, right? It has to do with the threshold mechanism of ROS. So ROS, it functions like a traditional morphogen. It's not necessarily in a gradient like a morphogen, but it does have different cellular outcomes depending on the level. So both too much and too little are very bad, right? You need a certain level of ROS for normal cellular homeostasis, and at very high levels, you trigger a protective apoptosis or cell death, right? But it's in the middle, which they call redox homeostasis, that you get these positive sort of active signaling and proliferation and cell differentiation um, effects, right? Specifically, this is required for stem cells, and it controls stem cell decisions, the level of ROS, right? The threshold level controls whether the stem cell is going to be quiescent and sort of stay the way it is, whether it's going to self-renew, whether it's going to differentiate, or whether it's going to undergo senescence or cellular aging. In pretty much every system that we have looked into date, ROS is required as an active driver of regenerative growth. So here are just three examples in Zinnimbus tails, in zebrafish fins, and axolotl tails. But in all of these, if you don't have this accumulation of reactive oxygen species, you do not regenerate. So it's a very attractive target. Not going to really talk about my model system much, but I have to show them because they're adorable. We do our work at the organismal level because our model system is planarians, and they are powerhouses of regeneration. They're very easy to care for. They're small. We keep them in Tupperware. They're adorable. They have a really weirdly large amount of adult stem cells. Up to a third of all of their adult cells are stem cells, whereas we have like probably less than 1% of all of our cells are stem cells. And they can do these weird things, like you can decapitate them like we've done here, and they go fine, and they just grow an entirely new brain and a new head, and they can do it in a couple weeks, right? Fortunately, they renew their pigment last, so it doesn't, we can sort of tell what the new tissue or this blastema is, which means it's a very good model system for studying wound repair, right? Because within about 2 weeks we can regenerate a whole worm, and then we can see what's going on. So we got into studying magnetic field effects under the radical pair mechanism, on the theory that the magnetic fields could be altering the amounts of the ROS, as we knew ROS was required for regeneration. So basically, the spin state will either lead to increased levels of ROS or decreased levels of ROS, right? This was first tested. I think this one actually is Frank Barnes' lab's work, or maybe that's the next one. In cell culture, where weak magnetic fields were able to affect cell differentiation. Here's slightly above geomagnetic, and we can increase the amount of cells that differentiate. Conversely, in like cancerous fibrosarcoma cells, if we're under geomagnetic, we can actually decrease the amount of cells that are regenerating. So a long time ago, we thought, hey, let's try it in planaria, because why not, right? And what we found is that even static magnetic fields, which is what we're working for, have different effects on the same biological process, in this case, head regeneration in planaria, depending on field strength. And it is not linear like a biologist wants out of a dose response. Um, but it does fit models, which I think I'm going to talk about. I don't know. We'll see where we are in time. But anyway, depending on the field strength you're at, right? When we cause an injury and we expose it to, so here the controls will be in the middle, right? So this is the amount of new tissue that grows normally. Um, we have several inhibitory field strengths. We take 200 as an example, and that prevents uh, tissue growth. And then we also have some strengths that are activating. So at like 500 microtesla, you can get way more tissue regenerating than normal. This is done at 3 days, which is sort of the max of regeneration for this model system, for new tissue at least. But if you back it up, this is an hour after injury. You get a bright accumulation of just general ROS species. This isn't specific, which weak magnetic fields can completely eliminate or completely upregulate, right? Depending on the field strength. And if we follow the dots from the 1 hour of ROS through the 3 days of new growth, we'll see that at these inhibitory and activation field strengths, we change gene expression that we know is required for stem cells to divide, right? Proliferation, so we can both inhibit and increase the amount of gene expression and cell division. And if you micromanage that, you actually see that it's both an increase in the or decrease in the actual number of stem cells, as well as increases or decreases, depending on field strength, in the progeny. So as the stem cells divide, they're going to mature. They're going to give daughter cells that are going to mature into differentiated tissues, right? So that was really exciting. Then we had a whole bunch of questions, right? So the first was all of our stuff was just general ROS without any idea what species it is. This work is a little frustratingly hard. We are working on ways to approach other things, but we looked at the easy stuff first. And at our 1 hour peak after injury, there is a peak of hydrogen peroxide at the wound, which is great. There is also an accumulation of superoxide. There is some at 1 hour, but there's a much larger peak at 2 hours, right? We really thought it would be hydrogen peroxide motivated, but we found that the weak magnetic fields, no matter kind of what strength we were at, really did not change hydrogen peroxide levels at all. And we tried a lot, and they don't. But we do regulate superoxide levels, right? So at 1 hour, we can start inhibiting superoxide accumulation, and we can't increase it though. But if we looked at 2 hours, we can definitely even increase at our activation field strength the amount of superoxide. So where we sort of left it was we sort of have a normal pathway of ROS through stem cell activities leading to new tissue growth. And we see that weak magnetic fields are affecting this pathway through superoxide signaling. And we are now looking at things like peroxynitrate, derivatives downstream of superoxide and that sort of thing. But what we're actually mostly concentrating on now is the what do I want to say? The bioelectro signaling part, because in other systems, it's well known that there is a relationship between membrane voltage and calcium and ROS in other cells, in other contexts, right? But it hasn't been looked at in regeneration, really. All right. And so I kind of left this in from another talk because I think it's also kind of interesting, because it also tells you what we're currently working on in our lab, but more importantly, it's sort of a biologist's viewpoint of what is important to know as the quantum biology field grows, right? So sort of the current challenges from a biologist's standpoint, not the physics or the instrumentation side, right? And the first one, of course, is it quantum? Because I always say, from a biological point, we have lots and lots of therapeutic treatments that we don't actually really know how they work. And if we can do enough controls and see the side effects and get it, somewhere, we can use weak magnetic fields as a therapy, even if it isn't quantum, right? If it's working. But we want to know how it's working because there aren't a lot of explanations that fit that if it isn't working quantumly. This paper actually just got accepted yesterday in PNAS Nexus, so we're very happy working with our collaborator, Christoph Simon, in his lab at the University of Calgary. Pretty exciting. This is kind of where I hope the quantum biology field is going, where the biologists are in real time, not like I do something and then theoretical biologists do something and then I read their thing and I try to do something. But we work together, right, at the same time. And so they started by wanting to model our superoxide data that we had published, but they had some questions, so they reached out. And then we realized it was kind of easier if we just, kind of worked together. And so they modeled our observations. They made some predictions. We told them that those predictions did not make any sense, but we would be happy to run the experiments to see what the real time experimental side suggested from their predictions, and it turns out their predictions were right, which was super cool because we do not know. This is sort of the first kind of validation that maybe the ROS is working from the radical pair mechanism because they modeled it on a radical pair mechanism, but we don't actually know what the radical pair is, right? But they were still able to, in a radical pair model, make a prediction that as an experimentalist, I was like, that's not how it's going to work, right? And as experimentalists, we often find out that our animals don't work the way we think they're going to work, which is why it's fun, right? So these kind of I think one of the challenges for the field is not just is it quantum, but getting how the interdisciplinary people work and working sort of together in real time is faster and more useful. All right, so the other thing we're interested in is what tissues it's affecting. So far, it's just proliferating tissues, but we need to know. So we're just looking at that. We are collaborating with other researchers. Currently, Dr. Sen at UNLV, who does vertebrate regeneration, and she's actually looking at hypomagnetic fields on the development and regeneration of xenopus. So we kind of want to know if these effects are conserved, because if it's just a planarian thing, interesting, but not really useful. And then the biggest thing here is sort of what's upstream and downstream of weak magnetic fields. And this is where the bioelectrical signaling comes in. So we are now looking at membrane voltage and calcium transients in relationship to both the ROS signaling endogenously and the effects of weak magnetic fields on these processes, right? So we find actually that at 200 microtesla, our data so far, this is preliminary, we need to repeat it a bunch more, but we seem to get an increase in membrane voltage where our current method, right? So our current data says 200 microtesla is going to inhibit ROS. And if membrane voltage is driving ROS, then it shouldn't be increasing that, except there is the feedback. So there's a lot of kind of technical epistatic things that we need to look at to be able to tease apart what's going on. But I think it's very exciting that we're seeing that the weak magnetic fields are affecting the parts of this proposed signaling pathway. And it's just going to take us figuring them out, right? We are also looking at what's downstream of weak magnetic fields. We have done RNA sequencing of both 200 microtesla inhibitory, the controls obviously at 45, the geomagnetic, and then the activation sort of 500 microtesla in 15 minutes, 45 minutes, and 2 hours after injury. And we have all of those data sets. The ROS accumulation starts as soon as 15 minutes. The stem cell proliferation peaks at 2 to 4 hours, which is why we kind of have this span here. So what we're finding is that we are pulling out downstream of weak magnetic fields a lot of ion channel genes, and many of them, like circa, the gene expression, are both endogenous sources as well as voltageous calcium channels, right? So this is probably going to feed into our feedback. There are a lot of neural genes that are also calcium related, so it makes a lot of sense, especially since regeneration is nerve dependent. Fortunately, we get a lot of ROS related genes, which is good. Cell cycle and stem cell genes, which is also good. And a very interesting pattern seeing that the biggest effects we're seeing with our inhibitory weak magnetic fields are early at like 15 minutes right after injury. So we're getting the biggest fields when we're blocking new growth right after injury, right? Conversely, when we're activating and we're causing more regeneration, we're seeing the biggest change in gene expression at this 2 hour time point, right? So there's a temporal component that's really interesting. And then I'm not going to go over this, but we're also trying to see if there's a difference between wound healing and actually regenerating new tissue. And we're finding that there are specific separate ROS mediated transcriptional programs. That paper is submitted and we're waiting for it. And then we're going to see how weak magnetic fields affect these different repair programs. So it all comes down to different effects of ROS on a threshold, which means when we are exposing them to different field strengths, right, we're going to get different effects depending on what time and what tissues that we are exposing them to. And if we can ever get EMF up in my lab, get the AC running, because right now we're just on direct current, I suspect low frequency EMF will increase the phenotypes we're seeing. It will magnify it because we have probably resonance with physiological like transients. All right, that is basically kind of my thoughts for the day. Thank you. I have a long history of a lot of great students who have helped with this, and a bunch of really awesome collaborators, including Frank Barnes, who got me started, and fortunately, some really nice funding from a bunch of awesome agencies like the NSF and the NIH that have allowed us to do this work.